Hey, very good afternoon. My name is Muslim and today I am going to give you a demo lecture for the purpose of faculty position of assistant professor of life science at IFS. So again, a very good afternoon, and today's topic for the life science topic for today's explanation is the replication of DNA. Replication of DNA is one of the most asked topic which is highly weighted in various exams like CSIR for their DBT as well as GATE or biotechnology as well as life science. So first of all, I will explain this topic in introductory positions. So replication is nothing but the process by which the DNA of an organism of a cell will get multiplied. Multiplied in the sense, the parent DNA will act as a template, and then the daughter DNA will get formed based on the template nature as well as the coding which is present in the parent DNA. So the nature of parent DNA will directly be act as a complementary in nature on which the daughter DNA will get formed. And based on this, the replication of DNA in prokaryotes is like the single circular DNA present in prokaryotic nucleus will get doubled, multiplied by 2 times, and then the amount of nucleic acid in the prokaryotic cell will be such that it can be then go, the cell can then go for the division, and it will form the 2 cells, 4 cells based on the type of mitosis is going on or the type of division the cell is going under. So replication of DNA. What is DNA? DNA is nucleic acid present in any cell. The basic one and highly stable one. Stable than the RNA, stable than the proteins, and highly conserved in the sense the nature of the DNA, its polypeptide chain sequence is highly conserved, and because it is highly conserved, the parental characteristics will get copied or sent to the next generation because of this nature of the nucleic acid's conservative nature. Conservation of the sequence, the polynucleotide chain is the basis for which the characteristics will get stably processed or given to the daughter, maybe daughter cells, daughter organism. And for this, the division of the nucleic acid or replication of the nucleic acid is highly important. And for which we are going to study the replication of DNA in prokaryotes. So prokaryotes, we know the prokaryotic cell, the simplest one having the single circular DNA, having the helical nature, double helical nature, forming a helix, as well as forming a circular structure. So this replication of DNA you will find in various references that it is explained on the highly in the structure model. So this is the highly explained model for the replication of DNA in E. coli. And another one is in Borrelia. So it is a genus of spirochete, and this is explained for the linear one, prokaryotic but in linear, and in E. coli this is given in the circular. So how circular I will explain this. So I think you understood what is replication. So we are going to just multiply the amount of DNA present, and that's what we are calling the replication of DNA, doubling of the DNA. So some basic concept you should know first, which will be highly significantly important, and I will let you know why they are important. So there is a rule in nucleic acids, we know. Basically we know it. deoxyhydrogen monophosphate will always get bound to deoxyhydrogen monophosphate, and it will form two hydrogen bonds. It will always form two hydrogen bonds. And deoxycyclic monophosphate will get bonded with deoxycholic monophosphate, and it will form three hydrogen bonds. So what's the significance? Two hydrogen bonds will be weaker than three hydrogen bonds, and this property is highly important in this initiation of replication of DNA, and I will explain you in short. So what's the significance is initiation of the DNA's replication, because high amount of energy will be required for breaking of this bond. We are going to break the hydrogen bonds with the help of an helicase enzyme in the starting of or initiation of DNA replication. But for two hydrogen bonds, less amount of energy will be required. The energy will be given with the by the help of ATP consumption by the enzyme helicase. So let's get forward. So another important term is semi-conservative nature of replication of DNA in prokaryotes. How semi-conservative? See here, the parent DNA, parent nucleic acid, the daughter nucleic acids. This is the point at which the replication is going on. It's called as replication fork. The replication fork. What's the significance of replication fork? Replication fork is nothing but the point where the amount of all the proteins, enzymes, and the whole polymerase tree will get attached at the replication fork, and this is the point at where the true activity of replication or multiplication of DNA is going on. And these are nothing but the daughter DNAs. Daughter DNA 1, daughter DNA 2. So this DNA is having a template which is coming directly from the parent DNA, as well as this is also following a template which is directly coming from the parent DNA, and one of the polypeptide chain of the helix of the daughter DNA is going to be formed with the help of polymerase 3 enzyme, DNA polymerase 3. And you can see on the both daughter DNAs, the amount of DNA that is going to get formed will be based on the polymerase 3 activity of enzyme polymerase 3, which will work based on the template nature of parent DNA. Okay. So now I will explain you the semi-conservative model. How we are going to like why I am calling this model semi-conservative. Why it is regarded as a semi-conservative in nature in various references, various books. Any book you will find it will be called as semi-conservative in nature. So why semi-conservative in nature? This is the point which will be get asked in CSIR, NET, DBT, JRF, as well as GATE Life Science, as well as GATE Biotechnology, Semiconductor nature. So there is a simple experiment, Michelson and Stahl experiment, highly asked in CS and GATE. So Michelson and Stahl experiment. Remember the name of the experiment? Michelson and Stahl experiment. This experiment gave the basis or the concluded that the semiconductivity of nature of replication of DNA and prokaryotes is there. So it is based on two theories. First is that there will be use of nitrogen 14 and nitrogen 15. This is this will form high molecular weight DNA. This will form low molecular weight DNA. Low molecular weight DNA. Why high molecular weight and low molecular weight DNA? Because it will have high atomic mass, low atomic mass. Okay. So the parent DNA, the parental cell of prokaryotic cell will be first the G0 or generation 0 will be grown in nitrogen 15. So remember I am explaining you the Michelson and Stahl experiment in which we got the basis of why we are calling it semiconductor nature of replication of DNA. So when G0 will be get replicated or the cell division will take place in nitrogen 15 medium, nitrogen 15 containing medium as nitrogen 15 will be the source of nitrogen. Or the medium will contain nitrogen 15 and so it will act as the source of nitrogen and so it will be consumed used by the cell such that the nitrogen present in nitrogenous bases will get nitrogen 15 will be get incorporated into that molecule of nitrogenous bases and we will get a nucleic acid which will be having high molecular weight. So nitrogen 15, this will form high molecular weight DNA. And after that, we will take the cell and for the parental or G0 generation 0 cells of the prokaryotes will take and it will be we will grow them in a medium or culture them in a medium containing nitrogen 14. And in generation 1, after completion of one growth cycle, we will get the proportion of cells containing the nitrogen 14 as well as nitrogen 15 will be shown based on the centrifugation process. So, and in copper chloride medium. So, centrifugation of the cell in the medium will give us two layers. In the tube of centrifugation tube, we will get two layers. First will be high molecular weight or high density DNA and the hybrid ones. Hybrid ones will contain nitrogen 14 from the medium, the nitrogen 15 from the parent. So half of the DNA will be or half of the cells will be containing nitrogen 15 only and it will form high molecular weight DNA and then half of them will be containing nitrogen 14 as well as nitrogen 15. Nitrogen 14 as well as nitrogen 15 and it will be having a hybrid nature of molecular weight. Hybrid in between light containing nitrogen 14 or high density or high molecular weight containing nitrogen 15 containing ones, and the mid ones will be the hybrid ones, which will be half of the proportion of in G1 generation 1 hybrid ones. And obviously in third generation what will happen? Generation 3, we are again going to take some cell or one cell or some cells, culture them in G3 generation 3 in nitrogen 14 containing only nitrogen medium. What will happen? What should we expect? The nitrogen 14 will be getting incorporated in nitrogen bases, and we are not getting nitrogen 15. So nitrogen 14 containing nitrogen bases will be more in nucleic acid. So more nitrogen 14 means the light or low molecular weight containing or low molecular weight having DNA will be increase in amount. So we can regard it as high amount of light DNA and low amount of hybrid ones. And lowest amount of high density ones. Lowest of high density ones. Okay, I think you got it. So this was basic instrument star experiment. You can get the pressure based on the time taken for the one generation to replicate multiply. The second one will be like the time taken for the amount increase. So there will be calculation based pressures too. So basic star experiment was based on centrifugation and based on molecular weight, based on the nature of nitrogen it will get incorporated. So next, so now I will explain you the bidirectional nature of the replication of DNA. So how bidirectional? So how bidirectional? So I made you remember single circular DNA experiment, prokaryotic cell, single circular DNA. Single circular DNA. What is this? The helical nature of single circular DNA. circular nature. How this DNA will be replicated? So there will be a point at which the replication of DNA will get initiated, and it is called as origin of replication. So there will be a point at which the replication of DNA will get started, and it will be called replication of DNA. And this point is regarded as noted as ORIC, origin of chromosomal replication. Origin of chromosomal. C stands for chromosomal. Origin of chromosomal replication. Understand the terms. Origin, the point at which the replication will start. Origin of replication. And then the proteins, the enzymes, the polymerase III, the most important enzyme, will get attached here. And it will start based on the helicase nature. The protein containing helicase nature will get attached at this point, and it will start the replication process. And there will be formation of replication forks. So these are replication forks. Which points are being called as replication forks? This is called as replication fork. This is called as replication fork. This was started at this. At this point, the initiation and this side was the point of origin of replication. The replication of DNA is going in this direction. In this direction. In this direction, it's going like it's going such that all the enzymes which will be having the activity of replication of DNA are moving on the parental template open polypeptide chain, and based on the nature of parental DNA, the DNA or the polynucleotide chain will get synthesized by the polymerase DNA polymerase III, and it will form again the daughter DNA. This is daughter chain. This is daughter chain 1, this is daughter chain 2. Okay. I think you got this points, the bidirectional nature. So how this replication of DNA will terminate? There will be also a point of termination. The point at which the two polymerase III enzymes will be about to collide. They are going in bidirectional in the opposite direction from the point of origin. And there will be a point where because it is a circular path, they are going to meet at some point. And this point is called point of termination. Point of termination. So, point of termination. After that, I will now let you know all the type of process, all the type of enzymes which will take part in this replication of DNA. And then I will let you know how the mechanism of replication of DNA takes place. So let's start with the names of enzymes. So you will get to know the work, the mechanism which the replication of DNA takes place. Proteins and enzymes. Why I am calling it proteins? Because some have like helping nature only and no enzymatic nature. Proteins and enzymes taking part in replication of DNA in prokaryotes. This is the point of discussion. Proteins and enzymes taking part in replication of DNA in prokaryotes. So first, remember the protein DNA. The first of the proteins which we get attached to the origin of replication. The point here, replication of DNA was going to start. The origin of replication. The point where the replication of DNA was going to start, it will be first detected by DNA based on the polynucleotide chain, the nature of polynucleotide chain, the specific type of sequence of polynucleotide chain present at origin of replication. This is so. What's this? Binds origin of replication. So second, DNA. B. Also a protein having enzymatic nature, helicase nature, will break down the helicase structure of the DNA. The helical structure of the DNA will break the helix of the DNA. How? By breaking hydrogen bonds. How helices will stabilize with the help of hydrogen bonds? Will break the bonds, the helix will be open. Third, DNA C. Helping nature. How will it help? How will it help? It will let the DNA B bind at DNA A. The position at where the DNA A is bind, the DNA C protein will help DNA B to bind at that position, open the helix. We will have to open the helix. We will have to take the each polynucleotide chain apart, on which there will be the positioning of polymerase 3s, and when the polymerase 3s will bind, we will start replication process, the mechanism of replication. Fourth, polymerase, DNA polymerase. There are 5 DNA polymerases. I will let you know for the sake of time. I will let you know 2 ones. DNA polymerase 3 and DNA polymerase 1. DNA polymerase 3 and DNA polymerase 1. Read this. I think you have got the point. Go ahead and करके let's start again. The fifth point, the fifth enzymes or topoisomerase. DNA topoisomerase. The enzyme having a lytic nature as well as DNA mediating nature. The enzyme, the only enzyme which can bind and which can break the phosphodiester bond. Phosphodiester bond will be here. Phosphodiester bond will be here. The polynucleotide chain is formed based on phosphodiester bonds. The phosphodiester bonds form between third hydroxy group of growing chain and incoming phosphate group of the triphosphate nature are forming the phosphodiester bonds, and these phosphodiester bonds are the backbone of helical nature of the DNA helix, and this will be break as well as form. This will be broke as well as form with the help of enzyme DNA topoisomerase. It can break the phosphodiester bond. It can also form phosphodiester. And the most important topoisomerase present in essential product, the DNA ligase. DNA ligase. So there will be primates. Remember this, DNA replication can't start directly by addition of deoxynucleotide phosphates. It can't start. It requires some growing chain having a third hydroxy group. A third hydroxy group. Third hydroxy group is required to start the process of DNA replication and addition of deoxynucleotide monophosphate addition to the growing chain. And we can't directly add deoxynucleotides, so there will be addition of a RNA. So in the first stage at which the DNA replication will get started, there will be addition of a small sequence of RNA, and it is called as RNA primer. So what is activity of DNA primase? Addition of RNA primer. Addition of RNA primer. I'll let you know the helicase. Once RNA primase will be add a small amount of RNA, DNA primase will add small amount of RNA. There will be a accepting group. Accepting group will be having a third hydroxy, 3' hydroxy group on the ribose. On the nitrogen base, and it will accept the phosphodiester bond from the fifth carbon of incoming deoxynucleotides. The incoming deoxynucleotides phosphate group at the 5' carbon on the ribose sugar, fifth carbon of ribose sugar. So helicase opens, helix, breaks, and join bonds. I think you have got it. You should read each point by stopping. Again, rehearse the points. I have told you processes and enzymes. So, origin of replication, breaking of helix, breaking of phosphodiester bonds, in polymerase activity. I think you have pretty much got it. Each point I have put. So, mechanism by which DNA replication will mechanism by which DNA replication will take place in prokaryotes. E. colon. So DNA polymerase 3 is the hero of replication of DNA. DNA polymerase 3 is hero of replication of DNA. Why hero? So now see structure of DNA polymerase 3. I am using different colors to make it interesting for you. So structure of DNA polymerase 3. This is tau complex, tau clamp complex. These are appendages. These are complexes containing alpha, epsilon and delta polypeptides. And this will be having, this will be containing the beta clamp. So I will let them name them beta clamp. The alpha, epsilon and delta complex. The alpha, epsilon and delta complex. And this is tau clamp complex. Tau clamp complex. So overall this is the holoenzyme, the DNA polymerase 3 holoenzyme. So it will be containing 3 DNA polymerases. It will be containing 3 DNA polymerases. This is a DNA polymerase. This will be 1, this will be 1 and this will be 1. So there will 2 strands of DNA will get formed after DNA gets differentiated or moved away. Okay. Then what will happen? One of the strands will move one of the strands will move through one of the beta complex, but one of the strands will move through two beta complexes, two DNA polymerase, 3 enzymes. Why? I will let you know. So two enzymes which will be active tau clamp complex will be two ones. First one, to open helix, the DNA helicase. And second one, to addition of primer, the RNA primer, the small polynucleotide chain of ribonucleic acid for the addition, for the starting of nucleic acid replication was RNA primer. It will be added with the name of DNA primers. So there will be also present of DNA primers. DNA primers. So when this will get, when the helix will get open, when the strands will separate, there will be addition of some proteins. These proteins are called as single stranded DNA binding proteins. There will be addition of single stranded DNA binding proteins. Single stranded DNA binding proteins. These will help the single stranded DNA to stabilize because the hydrogen bonds were open. The hydrogen which is present on these strands will be highly reactive. To stabilize them, there will be binding of single stranded DNA binding proteins. Got it? Refer to it. Yes. Okay. How the mechanism will go on now? Helix is open, single stranded binding protein is open, RNA DNA primers added, RNA primer go through, and now there will be addition of deoxypolynucleotides with the help of our DNA polymerase 3. And when this DNA polymerase 3 will act, first there will be formation of ribosome. At the origin of replication, all the proteins, I let you know DNA A, B, C, helicase nature. All the proteins which we find at the first at the origin of replication will be called together the complex will be called DNA chromosome. And all the proteins which we get bound to the replication fold with the DNA polymerase 3 will be called replication. So all this structure, all these proteins and enzymes together are called as replication. The whole structure. So when this will get moved, the single strand will move to DNA polymerase 3, there will be addition of deoxynucleoside monophosphates. And there will be two types of strands. There will be a lagging strand, there will be leading strand. When deoxynucleoside monophosphate will be continuously get added in the direction of 5 to 3, 5.2, 3 prime, continuously addition. This will take place in leading strand. This is parent, and this is the lagging strand, because it will not be able to add the mononucleotide phosphates continuously. The lagging strand, and because it will be lagging, there will be no continuous addition of nucleotides. There will be addition of after each 1000 nucleotide addition, sequence addition, there will be addition of some RNA primer. Again, on this RNA primer, there will be addition of some nucleotides, and then again there will be addition of the RNA primer, and it will form the Okazaki fragments. Each fragment, you can see this second fragment, this third fragment. These are Okazaki fragments. Okazaki fragments. So again, parent helicase activity, open the strands, DNA primers, addition of DNA primer, single stranded DNA binding proteins will sterilize the strand. The lagging strand will go through two DNA molecules, three molecules. First, it will be addition of single stranded DNA binding proteins. Then there will be addition of polynucleotides, but with a gap. Because of this, there is formation of Okazaki fragments. This is parent DNA. The parent DNA. DNA polymerase III complex. DNA polymerase III complex. The leading strand. The lagging strand. Lagging strand will contain Okazaki fragments. I hope you understood it. So after this DNA will get completely formed, it will reach the point of termination point, and then this two strand will get removed because of binding of protein TUS. Remember this foreign source protein TUS plus protein. Okay, got it? So what happened? There were the addition of DNA. There is still some presence of some RNA, some sequence of RNA. We will need to remove it. In Okazaki fragments, the amount of RNA is high. Each Okazaki fragment is containing RNA primer. This will be removed with the help of ribonuclease and DNA polymerase 1. DNA polymerase 1 or ribonuclease. You may get confused, but both have same activity, completely same activity. Removal of the first activity, removal of RNA primer. And second activity, if we remove the RNA primer, we need to again add some deoxynucleotide phosphate. It will be also done by only and only DNA polymerase 1, not ribonuclease. This will remove RNA primer and again add the deoxynucleotide phosphate. And this activity will be highly done for the Okazaki fragment containing lagging strand, and for the leading strand, only this first part, only the first part will be removed, and I can add deoxynucleotide phosphate. Okay. So after this circular DNA gets completely formed, the broadcast protein will be bound at point of termination. There are generally two problems concerned with this. Two problems. So what are the two problems? Formation of catenanes, and second problem is dimerized DNA. Catenanes formation. This is catenary. Intermediate deep circular DNA will be there. It will need to be cut at some point. To cut out this circular DNA should get away from each other. And again, this point there will be given a cut. And again, the cut will be one of the circles needs to be rejoined and will be done with the help of one of the topoisomerase. The solution to this problem is DNA topoisomerase. So this is another point which can get asked. You can see the previous question banks, in DBTs, JLabs, DBT, JLabs, assignment, GATE, life science. These questions are like frequently in the each year's question paper. This will give you a cut, remove, because of the cut, return. Then diagrams will be. What is diagonal here? At point of termination, the opposite strand got joined. This is like one daughter DNA, this is one circular daughter DNA, but they have got bound fused at the point of termination. So there will be enzyme recombinase XERCD. Remember that. This will help this binding, and then there will be formation of two independent daughter nucleic acids. So this is the solution for diagonal. Okay. I think you have got each point, and you will remember each point, the terms, semiconductor nature, bidirectional nature. Keep reading these points. Mechanisms, proteins, enzymes, member, mechanism, determinate nature, the addition of nucleotides, the addition of primer. These are some of the terms you should know and you should remember, and easily you can attempt questions based on replication of DNA. If you got these points, you have to read no nothing. This is everything. Okay, thank you so much.